Okay, hello everybody. So today's video is going to be about protein synthesis. And what protein synthesis is when inside your cell there's going to be some protein being made. So synthesis means to be made. And this happens inside our cells all the time. And let's see here. Okay, so and I'm going to explain protein synthesis from within the cell and happening in two different steps. Okay, hang on, let me get my board set up. Here we go. Okay, so the title of our notes today is protein synthesis. Oops, sorry. Okay, and so I'm going to break it down into three different steps. And so, first of all, protein synthesis is making protein. So let's write that down. And this is happening inside your cells, and this is happening all day, 24 hours a day. And so I'm going to break this down into three different steps. Okay, the first step I'm going to call transcription. Okay, so step one. We are calling it transcription. Okay, transcription. And so transcription happens inside the nucleus. So let's say happens inside the nucleus. And that's because the DNA is inside the nucleus. And so what needs to happen in this step of transcription is that one gene that is a segment of DNA needs to be copied. Okay, so one gene is basically a code for a protein, but step one is transcription, where that code of DNA is going to be copied into an mRNA. Okay, so the basic process of transcription is to copy a DNA gene into a mRNA. Okay, and so this is how it happens. Okay, so I'm going to write this out into like a story. We're going to draw a little bit. So the first step is that the DNA is split apart. Okay, so if you remember that we have a DNA piece, whoops, sorry. Let's draw a picture here. I'm going to draw a piece of DNA, so draw with me. Okay, and the first step is that the two strands of DNA are going to be separated. Okay, so here's one side of the DNA, and here's the other side, okay? And the two sides, the complementary sides, are going to be separated. And one side is going to be copied, okay? So let's say that we have a gene that starts here, and it goes all the way to here. And a typical gene, like we've talked about before, is about 1,000 nucleotides long, okay? Just on average, sometimes shorter, sometimes longer, of course. And so step one in transcription is that there's going to be this big enzyme that is going to bind to the DNA, and it's going to bind at this site that is called a promoter. And the enzyme that binds to the site that's called a promoter is called RNA polymerase. So we're going to write step one, RNA polymerase binds to the promoter. Okay, and let me just say a couple things here. RNA polymerase is an enzyme. Oops, sorry. This is a new whiteboard for me, and I'm having a little trouble getting used to it. No problem. RNA polymerase is an enzyme that makes RNA. Okay? And then the promoter is a section of DNA. that is right upstream of a gene. So a section of DNA at the beginning of a gene. At the beginning of a gene. Okay? So I'm going to draw the RNA polymerase. Let's see if I can switch colors here. I don't think I have colors. Okay. Oh, yes, I do. Okay, good. Okay, here's a red. Okay. So I'm going to say this is the RNA polymerase here, okay, in red. And so the RNA polymerase is a protein itself, and this is an enzyme that does chemical reactions like all enzymes do, and its job is to make RNA. Okay, so what's going to happen is the RNA polymerase finds the promoter, which is like right where a gene starts, and it's going to bind to the DNA at that point. Okay. So let's go back to black here. Okay, so step 1 is the RNA polymerase binds to the promoter on the DNA. And then step 2 is now the RNA polymerase is going to move along the DNA, and it's going to make a complementary piece of mRNA, which is basically a copy of that gene. Okay, so we have a gene here that's coded in the DNA, and now we're going to make a complementary piece of RNA that matches with that gene. Okay, so basically we're just making a copy of that gene. Okay, so RNA polymerase is going to move along the DNA and build a piece of RNA that is complementary to the DNA, and build a piece of mRNA that is complementary to the DNA. And what that means is that if you have an A in DNA, you will have a U in RNA. If you have a T in DNA, you'll have an A in RNA. If you have a C in DNA, you will have a G in RNA, and if you have a G in DNA, you will have a C in RNA. So it goes like this. So if you have, in your DNA, if you have, wait, why is it doing that? A T in DNA, in RNA, it's going to be a U. If it is an A in DNA, then you're going to have I'm sorry. Yeah. A, okay, wait, I'm sorry, how does it go? Oh, I'm sorry, A goes with A. Oh. If you have a T, I'm sorry, it's going to turn into an A in RNA. If you have an A in DNA, you will have a U in RNA. If you have a C in DNA, you will have a G in RNA. And if you have a G in DNA, you will have a C in RNA. So that's how it goes. So this RNA polymerase is going to move down the DNA, and it's going to be building a new piece of nucleic acid, which is called mRNA. So let's say, for example, okay, I'm going to move up here. Let's say, for example, we have a piece of DNA that is this. Okay, so let's say we have a DNA strand, and the nucleotide sequence is something like this. G, A, T, T, C, A, T, C, A, A. Okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's do one more. 8. Okay, so then the RNA polymerase is going to bind to this section. It's going to see that G, and then it's going to make a complementary piece of RNA, and so it's going to start with C because the DNA had a G, so the complementary piece is a C. Then when we have an A in DNA, it's going to be a U in RNA because there are no T's in RNA. And then if we have an A, we have a U. If we have a T, we're going to have an A. If we have a T, we're going to have an A. A C matches with the G. A matches with the U. T matches with an A. C goes with G. Then we have U, oops, U. Okay? So our complementary piece of mRNA, which is basically a copy of the gene, is going to be the complement to the DNA. Okay, so at this point, the RNA polymerase has built this new strand of mRNA. And so what happens is at the end of the gene, the mRNA is then going to be released inside the nucleus, and then it's going to go out of the nucleus into the cytoplasm of the cell. Okay, so I'm going to write that. This last step. 3 is once the gene is copied, the mRNA, whoops, leaves the nucleus and heads out to the cytoplasm. To the cytoplasm of the cell. Okay, so that is all that's involved in transcription, and all that's happening as the big picture is that there's a gene, which is a little section of DNA, it's a code, and that gets copied, and the copy itself is an mRNA, and now the mRNA leaves the nucleus, it goes through a nuclear pore, and it goes now is in the cytoplasm. And so we are now going into step 2 of protein synthesis. Okay, and step 2 is called modification of the mRNA. Okay, so modification of the mRNA. Okay, so we are now in the cytoplasm of the cell. And so there are going to be 3 things that happen to this mRNA, okay, for it to get ready to go into the last step of protein synthesis. And what the first thing that happens is that we have this long string of RNA, which is basically a copy of that gene, and there's going to be a little cap that gets put on the 5' end of the mRNA. So the first thing that happens is there is a methylated cap put on the 5' end. Okay, and so that's like the bottom, the top of the strand. Okay, at the top of the strand there's this little cap. So let's say we have this mRNA. I'm just going to draw it as a straight line. But I want you to imagine that in this line there are lots of nucleotides, so A's and U's and C's and G's. And so the first thing that's going to happen is there's going to be this little cap put on the end, and what this does is it is going to protect that end because this mRNA is very fragile and it can fall apart very easily. Okay, it's only meant to be used temporarily, so it doesn't need to be robust, and it isn't. But there's this little cap put on it just to protect it from falling apart. Okay, the second thing that happens is that this transcript, which is made from transcription, it's a piece of mRNA, is made up of nucleotides, and parts of this transcript can be cut out and then be glued back together in all sorts of different ways, okay? And this is what's so amazing about our genes is that one transcript can be used to make lots of different proteins, okay? So I'm just gonna put some lines here. So let's say from here to here is a section, from here to here is a section. And so this transcript can be broken down into different sections. And so some sections are called exons and some sections are called introns. So then we have an exon and an intron. Let's say we have 2 introns in a row, and then an exon and an intron and an exon. And let's say exon. And so what's going to happen in this step 2 is that all the introns are going to be cut out with enzymes that are like little scissors and removed. And then all the exons are then going to be glued back together. Okay, so I'm going to say that introns are cut and removed. And exons are glued together. Okay, and so this is again very very complex and really efficient, because like I said, the same transcript can be used to make lots of different proteins, because you can cut and paste them all in a whole bunch of different ways, and that's just really awesome. So for example, what we're going to have now is we have our little methylated cap, and then I'm going to have the transcript, and it's a lot shorter now because all these exons are cut out. So this intron is cut out, this intron is cut, and so all we have left are the exons. Okay, so now all we have are just these exons that we had before. All the introns are gone. And so now our transcript is shorter and basically is a different sequence than it was originally. Okay, so then what's going to happen is step 3, and the third thing that happens to modify and get our transcript ready is that we are going to put what's called a poly A tail at the other end, at the 3' end of the transcript. So we're going to put poly A tail at the 3' end. Okay, and so what we have now is we have our methylated cap, okay, and then we have our shorter transcript, and then we're just going to tack on a bunch of A's at the end. This is called a poly A tail. And what this done again is protects the other end. Okay, so one end is protected by a little cap, and then the other end has all these A's, and they do start to degrade, and it's okay because the A's don't do anything. Okay, the A's are just there as like a buffer so that if degradation happens, no problem. Okay, so now our mRNA is now modified, and now it can be used for step 3. And so step 3 in protein synthesis is called translation. Whoops. Okay. What happened to my whiteboard? Oh my goodness, what happened here? Okay. Step 3. This is called translation. Okay, and so we've talked about this several times already in other videos, that proteins are made on ribosomes. And so now this step is going to be done on a ribosome. So this happens on a ribosome. And we've talked about ribosomes before, and there are 2 kinds of ribosomes. There are some that are free-floating in the cytoplasm, and there are some that are stuck to the endoplasmic reticulum. And so depending on what the protein is going to be used for determines which ribosome it's going to be made on. And so I'm going to again draw a picture and make a list of what's happening in translation. So the first thing that happens in translation is that we have our modified mRNA. It binds to a ribosome. Okay, so we need 3 things to start translation. We need an mRNA, a ribosome, and we need this other RNA molecule called a tRNA. Okay, so those 3 things need to come together and bind to initiate this last step of protein synthesis called translation. And so I'm going to draw this. So we have a ribosome, which, if you remember, is made out of 2 subunits. Okay, so there's a large subunit and a small subunit of the ribosome. Okay, so this is the ribosome. Let me just label this here. Okay, and the ribosome is going to bind to the mRNA. So here's a little cap, and then I'm just going to draw this line. This is the mRNA, this is our transcript, and the ribosome is essentially going to read this mRNA, and it's going to read the mRNA 3 nucleotides at a time. And 3 nucleotides on an RNA is called a codon. Okay, so I'm going to write this down. A codon is 3 nucleotides in a row on an mRNA. Okay? And so the ribosome is going to read the mRNA one codon at a time. And so let's say we have this first three here. Okay, so these three are the first three nucleotides on an mRNA, and it's going to be a U, A, C. Okay, and so this ribosome is going to find these first three nucleotides on the mRNA. It's going to say, oh, it's UAC. So what it's going to do is it's going to grab a tRNA and it's going to try to find a match to the first codon. Okay, and so let me just step aside for a second and talk about what a tRNA is. Okay, so a tRNA, which stands for transfer RNA, is a piece of RNA, and what it looks like is it has this like four-leaf clover look, and then it's carrying an amino acid. So this is an amino acid here, okay? And it's in this particular shape because it's made of nucleotides and they bind to each other to make this three-dimensional shape. And it has three nucleotides off the bottom called an anticodon. This is the anticodon. And so what the ribosome is going to do is going to try out all these different tRNAs to find the matching anticodon to the codon. Codon being on the mRNA, anticodon being on the tRNA. So what's going to happen is this ribosome here is going to find a tRNA that matches. So it's going to find a tRNA. It's just going to try out a whole bunch of different tRNAs, tries one, and just over and over and over until it finds a match. So it needs a tRNA that's going to have an A here, a U here, and a G here. Okay, so it's going to have an anticodon that is a complement to the codon. And once it finds the match, it's excited. It's like, yes, this is it. So what's going to happen is this is going to be the first amino acid in the new protein that's being made. Okay, so once it matches, like, yep, this is the right one, I'm keeping this protein. Okay, so step one of translation is the ribosome is going to find the matching tRNA to the first codon on the mRNA, and then it's going to do this all inside the ribosome. So the ribosome is like this little machine, and all it's doing is it's matching the anticodon to the codon. Okay, so once it matches the first one, then it's going to try to find the second one. So step two is the ribosome. Okay, so first, the step one was three things need to happen. mRNA, tRNA, ribosome all come together, okay? So the ribosome will then match the second codon. Remember, the codon is on the mRNA. So second codon to a new tRNA, Okay, so I'm going to come back up here to our picture so I could draw it. And then let's say the next codon is here. Okay, and I'm just going to say that it's AGG. Okay, is the second codon. Remember, a codon is 3 at a time. So then this ribosome is going to just search all the tRNAs that are hanging around and try to find another one that matches this second codon. So it needs to find one that has a UCC. That's the anticodon. So UCC, and once it finds that match, it says, yay, I found the match, and it's going to keep this amino acid. So at this point, what's going to happen is the ribosome is going to bind these two amino acids together. And then this tRNA, the first tRNA, is going to leave the nucleus, but this the first amino acid is going to stay with the second amino acid. Okay, so let me write that down. Okay, once there is a match, the first amino acid stays in the ribosome and binds to the second amino acid. And binds to the second amino acid. Okay, the tRNA, the first tRNA, which no longer has an amino acid bound to it, is going to leave the ribosome. The first tRNA leaves the ribosome. Okay, so let me draw what that looks like. I'm gonna scoot up here a little bit. Okay, so we have our ribosome, and then our ribosome has two pieces. We have the methylated cap, we have the mRNA, and the first codon has already been read, the second codon has also been read, and we have our tRNA here with the second amino acid, and the first amino acid is bound to the second one, and they're stuck together. Okay, a peptide bond has been formed between the two. Okay, then what's going to happen is that ribosome is going to move down and it's going to read the third codon. Okay, and it's going to find the matching tRNA for the third codon. So I'm going to say step 4 is the ribosome moves down the mRNA and finds the matching tRNA to the third codon. Okay? Moves down there. Okay, so what's happening in this picture now is we have the ribosome moves down and then finds another tRNA that matches the third codon. And boom, bingo, found it. So what's going to happen next is this second tRNA is going to leave. This amino acid is going to bind to this amino acid, and now we're going to have 3 amino acids in a row. Okay? So let's write that down. The third amino acid is added to the chain of amino acids. The third amino acid is added to the chain of growing amino acids. And this continues. So the ribosome is going to keep moving down the mRNA, adding amino acids to each other. The tRNAs are delivering the amino acids one at a time, and the ribosome goes all the way down until it hits what's called a stop codon. Okay, so I'm going to say ribosome continues to add amino acids to the growing chain, continues to add amino acids to the growing chain until it reaches a stop codon. Okay, and at which point the polypeptide chain or the protein is now completely made. Okay, and so once the ribosome hits what's called a stop codon, it stops translating. And it lets the polypeptide chain go, and that string of amino acids is folded up into a 3-dimensional shape to make the protein that it's supposed to be. Okay? And so maybe you're wondering, like, how do we know which amino acid goes with which codon? And so I'm glad you asked that. Because what we do is we use what is called a genetic code. And so I would like to show you how to do this. Okay, so that's it for translation. I want to work through one problem with you, okay, to help you with the questions that you're going to be asked. And so I'm just going to write down a DNA sequence, and then we're going to transcribe that into an mRNA sequence, and then we're going to translate that into a protein sequence. Okay, so we're just going to do a little practice here to show you how to do something like this. Okay, so let's say we're going to do A, T, G, G, A, T, T, C. Okay, so let's say this is our DNA sequence. And so we're just going to say this is our gene. So this is DNA. Okay, and this is the gene that we have. And so when you have a gene, it's in the nucleus and it needs to be transcribed. And so what the cell is going to do is it's going to make it into an mRNA. So to do that, you would do the complement, and remember RNA has U's instead of T's, so it has a uracil instead of thymines, okay? So we're going to have a U, A, C, C, U, U, A, G, okay? So then what we want to do is we want to put together an amino acid sequence. And to do that, you have to use what's called the genetic code. And let me pull that up for you. Let's see here. Hang on a second. Right here. Genetic code is here. Okay. So here's the genetic code. And I just want to put this off here to the side. And so to look at the genetic code, what you need to do is act like a ribosome. And a ribosome is going to look at the first 3 nucleotides in the mRNA, which is the first codon. And then there's a second codon, and then there's a third codon. Okay, so the ribosome is reading the mRNA 3 nucleotides in a row, 1 codon at a time. Okay, and so it's going to look at the UAC. So what you need to do is look at this genetic code, and you need to find the UAC. Okay, so this table here are all the possible codons, like all the possible combination of 3 nucleotides, okay? And so this is all that's possible. So in this table, if you look on the left here, this is the first letter. So if the first letter starts with U, it's going to be in this row. If the first letter is C, it's going to be here. If it's the first letter is A, here, G, down here. And then up here is the second letter, and over to the right is the third letter. So you're going to find UAC in this table. So UAC is right here, okay? And so and then you look over to the right, and this is the code. This is like an abbreviation for the amino acid. So it says PHE. So the amino acid for UAC is PHE. Oops. I'm sorry, hold on. PHE, which stands for phenylalanine. Okay, then you look at the second codon, which is CUU. So we're going to look at CUU. It's right here in the genetic code, and the amino acid is LEU. That stands for leucine. So second amino. And then you look at the third codon. It's AAG. So you look back at this code. AAG is right here. The amino acid is LYS, lysine. Okay, and so our amino acid sequence for this DNA is going to be phenylalanine is the first amino acid. Leucine is the second amino acid, and lysine is the third amino acid. And you can go on and on like this. And so that's how we do it. So I just want to point out one last thing is if you look at the genetic code, you can see that 3 of these are in bold, and those are the stop codons that I mentioned before in translation. So the ribosome is going to go through and read all the different codons until it reaches one of these 3, UAA, UAG, or UGA. Once it hits one of those, it stops, releases that polypeptide chain, and now the protein folds up automatically, and it starts to do its thing. Okay, so I hope you enjoyed that video, and you learned about protein synthesis, and I hope you're all having a really good week. Thank you, and have a good day. See you next time.